Thank you for inviting me to give this presentation. I'm really honored and I'm really pleased. So I hope you enjoy it as much as I do. And it has been absolutely amazing for me to work in this discipline, which I just didn't know. So I'm not a gastroenterologist. I'm just a dumb little neurologist. So bear with me. What I will talk today about is about the gut-brain axis in epilepsy and cognitive dysfunction, which we found some amazing results, which you will see later, which I'm still, a couple of them really challenged my mind, and I hope it will go similar to you. So at the moment, obviously, I have some disclosures to make. All of the opinions obviously are mine and our research. And then I also have obviously some funding, which I'm very glad and I'm really grateful for all the funding we do. But you obviously have to appreciate that when I'm speaking. Well, there's no presentation where I don't start with this beautiful, beautiful creature. And she was called Beauty. Unfortunately, she was called Beauty. She started to have epilepsy when she was 4 years of age. I adopted her from a shelter, and then I had her until she was 17 years of age. And I learned so much from her, not only what epilepsy means, but as you can see in the next video, also what cognitive dysfunction means. Yeah, this beautiful old dog just staring, being wise there, and all those youngsters, they're the other dogs we had, you know, and in a second she's sniffing on some flowers. You know, it's called wisdom when you get older, right? When you're young, you're just a bit woo. Yeah, so she had epilepsy for 17 years, and a lot of the things I learned, and you think like, wow, I didn't realize. And one of it, you won't believe it, is that she also often had gastrointestinal problems. And I just didn't think about it. And I thought, well, this has some irisetta and have some issues with that. So you will see there's a lot one can learn from their own pets, as most of you will agree. When we started to look into what can we do with changing the diet, it came mainly out of frustrations, because the bottom line is, you know, there's a lot of new anti-epileptic drugs, same in humans. The one big change has been that there might be better in side effect profile, but in normally otherwise they didn't really bring the oomph. They didn't really bring the bettering of the clinical signs, which are the seizures in epilepsy. So we looked at new ways of treating a very difficult to comprehend disease. And we were looking more at a multimodal approach. So obviously drugs being an important one, but also looking, could we potentially change the diet? Because when you speak to clients, a lot of them have hypersensitivity issues and other things. And then you just change the diet, for example, to put them on a less hypersensitive food, and then they get better. And we're like, oh, that's really interesting. Why is this? And then we were looking more into the gut-brain axis, or brain-gut axis, because I'm obviously a neurologist. And there's so much out there. People have shown you can change bidirectionally, on the one hand side via the vagal nerve. So a lot of information, and you see that later. The vagal nerve is, I think, more important than we ever thought. And we use this even in epilepsy. We stimulate the vagal nerve. We change the rhythm of the vagal nerve. And by doing this, we reprogram the brain and also improve actually inflammatory responses in the brain and also the seizure activity. We know that you obviously have digestion, and then you get stuff which goes into your bloodstream, then normally bypasses the liver, gets metabolized or detoxified, and then goes into in the circulation, and then it has to pass the blood-brain barrier, right? So we know that certain substances can pass and others not. We know there is a thing about the inflammatory pathways as well, which can change also how the brain is working. And we have seen that changing just the microbes by doing the gut microbiota with an antibiotic can change already response to anti-epileptic, and I will show you that as well. We also have shown that stress can influence the gut and the microbiome, and vice versa as well. So there is a lot of crosstalk. So how can we use this, and what is known? So the first thing is, no surprise, and I put all the papers on the top with a little QR code, which you can later perhaps or now take. You know, we did this study, and people said like, oh, what are you doing there? And this is actually the most successful study we have done in terms of seizure control, because we selected a lot of dogs, and we asked the owners of dogs which had very common seizures, like once a week or every second week, to collect saliva, so we can measure their stress response to the seizure activity. The same also for the owners. And what happens then, like always does, right? When you do those trials, suddenly none of the dogs had any more seizures, so we had to recruit more. So it was obviously the classic thing of a placebo effect or whatever you want to call it, right? But saying this, no surprise, no surprise, right? When you have a seizure, you will get a stress response, right? That's in the owner and in the pet. So that's not a surprise. You can't. Seizure is stressful, so you do this. However, what's interesting then, we looked at more the chronic stress level, right? We looked at what happens when you take care and look more at how the chronic stress levels are. Is there also a change? And what we found there was that there actually was a decrease in the stress response. So what we suggested then was that they actually have like a chronic disease process going on, and because of that, there is actually like a chronic fatigue syndrome of those stress hormones. So something is already going there and modulating the pathways there. And then this study came out. Fascinating, I think, right? Because you all know this because you are vets, right? If an owner is neurotic, their dog is neurotic, right? If an owner is stressed, the dog is stressed. And now, I mean, some of you might know that we did a lot of corona sniffer dog work. Dogs can sniff adrenaline, they can sniff cortisol. So they know exactly what is going on. So as more stressed we are, the dog is more stressed. As more stressed you are, as more gastrointestinal signs you might have, as I'm sure most of you are aware, right? So there's a big interlink between the stress of the owners and also the pet. And we looked into this a bit more in detail and found that obviously, owners find the disease really stressful. They can't leave the house. They're really they can't go on holiday and so on. And we titled, it was a qualitative research, so we titled the presentation as "We Have a Ticking Time Bomb." Yeah, that's what someone said to us in this qualitative research. So there's definitely a lot of stress. And what we do now when we treat epilepsy, we do look at stress management, not only of the pet, but especially of the owner. So similar like with childcare, right? So if you are relaxed as a parent, also your pets are relaxed or your child. All right. And it is stressful when you look at this, right? They have a seizure, right? But everyone always focused mainly on those epileptic seizures. But the epileptic seizures are not covered in mnemonic disease because there's millions of reasons why you can have epileptic seizures, right? The thing is, what happens in between the seizures might be even more important. And we know this from human medicine, that there's a lot going in between. So I told you already about a little bit about stress, but there are also other factors, because we know nowadays, in the beginning, no one believed us, that between the seizures, dogs are different as well. With idiopathic epilepsy, that means that you didn't find a cause which you could identify with our means at the moment, blood work, CSF, and also obviously MRI scan. They still are not normal, the brains. Yeah, they have a brain disease. Yeah, epilepsy is a brain disease. And what can happen when you ask people with epilepsy, what else apart from the seizure activity is epilepsy for you? Then you suddenly find there's things like depression, anxiety, and confusion, and so on, ADHD-like behaviors. And there were good times when you were a researcher. So it was not like Jan Sukodowski sitting in a lab with pipettes. What you had was you were sitting in the sun and eating some olives and drinking some red wine. And at that moment of time, what people were finding is that melancholics become ordinary epileptics, and epileptics become melancholics. And this is something which has been found again in our times, that people with a depression or anxiety have a higher likelihood of having actually epilepsy. People with epilepsy have a higher likelihood of having depression, anxiety. So we were really intrigued by this, and we wanted to see if the same actually happens in dogs, right? And we have a brain gym at the RVC, and what we found is that you could actually see that separation anxiety was increased in the dogs which had epilepsy compared to controls. We also looked into this with a questionnaire, which was before we had the brain gym, and again we found that 70%. So that's quite a lot, right? This is more than 2/3 of the dogs with epilepsy have changes in behavior, right? They have more fear and anxiety type of disorders, and especially when it comes to stranger-directed fear or other pets-directed fear. Yeah. And the other thing is, what was also very interesting, I'll show you the original study, is that we found that they have more ADHD-like disorders, right? That doesn't mean that dogs have ADHD. I just want to make this clear, but like behavior, they have problem with their impulsivity control. They can't concentrate as well. And we have shown this as well for dogs with epilepsy. And it becomes really important later when we look at how we can use poop treatment strategy. Yeah. And this is the beautiful Tarja Jokinen. She did from Finland. She done an amazing research. What she did is she looked in those beautiful dogs up in the right corner. These are called Lagotto Romagnolus. Some of you might know these dogs, and they have what is called a benign childhood epilepsy. So they have a gene defect, and there's another gene which will compensate for this gene defect later on in life. So they grow out of the epilepsy. They grow out of having epileptic seizures. But here's the thing, when she looked at them 5-6 years down the line, they didn't have any more seizures, but they still had behavior change. And in a beautiful work, what she did, because she looked into what actually changed, and she found exactly the same as we, because often when you find something, obviously, you don't know if it's true or not, right? But she found that they could not concentrate, they had inattention, and they had hyperactivity, impulsivity control issues, and no seizure activity. Really interesting. So something changes in their brain, right? And then the other question is, when does normal become abnormal? I'm at the moment at the London Vet Show, right? And back in the turf, so to say. And at the RVC, I did obviously change, right? And the question there is, did I actually also cognitively declined as well? So I'm a person, I feel still like 22 or 23 now. Perhaps I used to be 18. Now I have like feeling of 22, 23. A lot of energy in life, and what's happening is that somehow, obviously, I aged outside, right? And we look at the mirror and like, ooh, that looks very different. And especially when you look at pictures, and when you look at your cognition, right? I think, knowing that forgetfulness is something which happens, right? I always lose my keys, and then my wife tells me that they should be on their place, and then I get really mad because then I don't find the keys. So that's not the right moment to tell me they should be on their place, right? But that's not abnormal, right? We some of us are very good in remembering where the keys are, and others not. That's not necessarily abnormal. If I find my keys in the fridge, then it's a different story, right? And what is it with cognition in dogs? And there's a big stigma in people with epilepsy, because people perceive that they have they're a little bit impaired in their intelligence, and that's actually not true. That's not true. However, if they have also a cognitive decline, it will be aggravated by the epilepsy, right? So a colleague of mine, a postdoc of mine, she had epilepsy. She's the smartest person. She's definitely brighter than the sun and brighter than me. So we wanted to see if this also is something happening in dogs, and like I said, the thing is, what epilepsy might do, it would just aggravate your cognitive dysfunction. And when we were looking at, we did a questionnaire. We had like 4,500 dogs included. It was just adult dogs. And there were two things which featured. Two things. One, obviously, cognitive dysfunction. Yeah. So if you're older than 12, a dog, 1 in 3 out of the dogs will have cognitive dysfunction. If you are 15 to 16, 2 out of 3 dogs, right? And the same was with epilepsy. So dogs with idiopathic epilepsy had a change in trainability and also cognitive dysfunction score. We've shown that when you have positive reinforcement, so when I tell you, hey, great, well done, and I give you some candy and I reinforce the learning what you have done today, then you might have a more protective brain. So a lot of us staying active, and you are watching and perhaps rewatching this webinar, will be great, right? And there's obviously certain drugs which can also affect it, like zonisamide and potassium bromide. We looked more in detail what actually changes in dogs, and it's mainly, and that's not a surprise, it's mainly the spatial memory which is changed in these dogs. It's not so much a problem solving, more the spatial memory. Yeah, and when you look actually why this might be, it's actually really simple. We have a structure called the hippocampus, and in the hippocampus area is where your memory retrieval is. So spatial memory is, for example, in the hippocampus. Often lateralization between left and right, and then there's also some recall you do, but also for memory forming, right? So it's a structure of the hippocampus where a lot of information comes in, gets processed, and goes out again, and vice versa. Okay, and guess what happens? It's an area which is prone to be harmed by the epileptic seizures, right? And this is the dog where you can see. Hope you can see my mouse here. Had a little bit of a swelling, had a status epilepticus, especially on one side, and then a couple of years later, this brain tissue was gone, and this is the piriform lobes. This is part of the limbic system, and limbic system might say something to you. That's not only where the hippocampus is connected to, but also where stuff like feeling emotion, the amygdala, fear and anxiety. So you can see this is an area which is really compacted, and it can explain why cognitive decline can happen in these dogs between the seizures, and also the behavior changes. It makes sense, fear and anxiety. So that makes perfectly sense, right? So now we have a problem, right? Because the drugs we are giving, if you have phenobarb as a person, you are more sedated, you are sleeping. It was developed as a sleeping medication, right? So this is not really harming. So how do you go about changing something in the behavior and also in changing something in the cognition? And when you look into this, and this is where it really becomes quite interesting. We know that when you have an epileptic seizure, the area which I just shown you will become more glucose metabolizing, right? The seizure, a lot of energy you need, glucose will give you the thing. However, when you do between the seizure activity, you will have less activity. You will have less metabolism of glucose. And people have even shown that the mitochondria in these areas will also have a problem. And you can see this very nicely on this paper from Daisuke here. And it's unilateral, like the case I've shown you before. And you can see how blue it is. So it shows you that probably you have an energy change, how you can use glucose, and you don't use enough. You have a hypometabolism of these glucose in this area. Fascinating, right? And that can explain you potentially why you have more anxiousness and also cognitive deficits. So we know that glucose is obviously important. We know that the brain is important. Everyone always tells you that your brain needs sugar, yeah, and 20% of your glucose you will take in your brain, right? But we also know that, and that's actually based on a lot of research from Gary Pan from Purina, we know that actually you have a change in brain metabolism when you get older as a dog and as a person as well. Your glucose metabolism, very similar to epilepsy, your mitochondria might not function so well, and your glucose metabolism, especially in the neurons, is not functioning as well. And this is where it's interesting, because it's one of those dogma which has changed. We now know that actually your brain, the neurons work fantastically on ketone bodies, especially when you get older. The same happens in epilepsy, where people have described for a long time that people who were starving, who had then the ketone overproduction, actually a better seizure control, right? And then they were playing around with diets to mimic this, right? So the guys who still need, when you get older, glucose are your glia cells, but these neurons are 4 to 6 times more efficient on ketone bodies, right? And that led us, because we knew from the work been done about cognitive dysfunction, and that dogs with cognitive dysfunction, or with brain aging, to be correct, improved with a more ketone-producing diet, which were medium-chain triglycerides. Medium-chain triglycerides are C8 to C12 and mainly C10 to C12, which get fastly metabolized and easily metabolized into ketone body. which is ideal for the dog because, as you know, it's very difficult to get into ketosis. So you have MCTs to improve the ketone levels in the blood. And we've shown this now in one study, the first one, where 14% of the dogs which were drug resistant to phenobarbital and some of them also to potassium bromide then became seizure free. And half of them had more than 50% reduction, which is kind of the former definition of classical drug-resistant drug response. And this is very similar. If I would have used a third antiepileptic drug, I would have got very similar number, which is very interesting indeed, right? And we have shown this now in another study with MCTs where we did just adding an oil, and we have shown it also in the field study. So that is quite a robust. It doesn't mean that all the dogs, but around 25% so do respond to MCT diets. And the interesting thing is, and this comes back to what we just said earlier, apart from the seizures where we always look at, also their ADHD-like behavior improved. It was not necessarily always the same dogs, and also some of them had a reduction in impulsivity control, so better impulsivity control, so also less ADHD-like clinical signs. And guess what? We also looked at cognition, and cognition also improved, right? The dogs had a 20% improvement in trainability, spatial memory, and problem solving. Like I said before, it's not as much affected. So there was only, depending if that's clinical significant, right? But things over 20%, you normally see in a clinics that they do make a difference. And when you look into how drugs work, we have two main pathways, but the one pathway we mainly use, right? So I spoke to you about changing how energy levels are used. Now I will speak to you a little bit more, because MCTs are actually doing something else, which is quite cool, because they also have activity on NMDA receptors. Because most of the drugs we use in epilepsy treatment are just on the GABAergic side, right? So there are some there's a patent out there where people have used certain C10s which do block glutamatergic, so the excitatory pathways. So the diet shows you can not only improve seizure control, behavior, and cognition in some dogs, but also give you a reason not only by glucose metabolism, so alternate energy sources, but also by giving input perhaps in these some dogs where it works on the AMPA receptor, and that explains why it works in some dogs and others not. So if you can't sleep at night, we summarized in this article all the different types of functions of how MCTs could work or not work, and we discussed it. I think one of the things which I would like to highlight, because we will talk about this in a second, we could also see there was shiftings in GABA and glutamate concentration. So the excitatory neurotransmitter is glutamate, which decreased, and GABA increased. And you'll see later why this is important. Because we also looked at the feces, and we did this obviously with Jan, who's definitely more knowledgeable about this than we will ever be. And we're really grateful for this collaboration, which has been very, very fruitful. We didn't find huge changes, and as Jan would go into, often you don't find huge shifts in diversity patterns. But what we did find a couple of changes in single bacterial strains, which then what you do a little bit like when you do gene research, you look what other people have reported about them and see if you can retrofit why something might have happened. So we definitely found that Bacteroides it increased, and that was with the MCT consumption, and people have shown that they might have a decrease in aggressiveness in dogs. What we didn't find, you might say, why did they look at Akkermansia were not detected in the dog? And at least that's what Jan says, because Akkermansia might be in the crypt and not being shedded outside, was that they have been very featured in this one. And this is really interesting because that changed a lot in the epilepsy research and has been heavily debated as well. So what those guys did in this Cell paper, they had rodents which responded to a ketogenic diet and rodents which didn't respond to a ketogenic diet, right? And then what they did is the ones which responded they took their feces and gave it to the rodents which did not respond to the ketogenic diet. And suddenly they could transfer the seizure controlling effect, right? It was actually possible to control suddenly the seizures by just giving the fecal transplant. And then what they also could find is that it was Akkermansia and Parabacteroides. And that's why the Akkermansia we looked at, which were changed. And then they did something, obviously, what you could not do in other settings. They looked even at the concentration in the brain and found there were changes and increase in GABAergic compounds in the brain, right? So GABA was increased. So they said, probably what these bacteria do, and that probably is the causality. So they could transplant the seizure response, which is fascinating. Yeah. Then other people have looked into other factors. A really nice study from Karen Mugnana. Lactobacillus, certain strains have been also associated in people with epilepsy to be different between drug responders and not. And she looked at a twin study, so to say, looking at not true twins, but they were dogs which were living in the same household, and she looked at dogs which had epilepsy and other dogs without, and saw if there was a difference, but they couldn't really find anything major there. There is a study where they looked at 10 epileptic dogs and 12 healthy beagles, and they did find some changes, which also made a little bit of sense and could also explain why GABA could increase. One has to be a little bit careful because they were just beagle dogs and the other one were mixed dogs and the different types of setting. But there's some evidence that the microbiome might be changed in epileptic patients. There's another study by Jan, which they've done in cases of an inflammatory brain disease, where they found also an abundance of Prevotella C, and again, then they looked obviously at the literature before and could see that the inflammatory responses are changed and said like, that's really interesting. And they found a reduced abundance in short-chain fatty acid producing bacteria, which becomes important again in a second as well. So we wanted to say, okay, let's start in the beginning, right? Let's take dogs which should be treated with phenobarbital, so they have idiopathic epilepsy being diagnosed, and we want to see what do actually the drugs do. So start with one step after the other, yeah. So because most of these other studies they used a mixed, they had like epileptic dogs, some of them treated, non-treated, and we wanted to see. Let's first look what does actually the drugs do. How do the drugs work? How does actually phenobarbital does? And there were a couple of things which I was amazed by because I didn't expect it. Yeah. So we did a classic, diagnosed them, started it, and then there were 90 days on the drug, and then we took another poo sample. We looked at, did some behavior testing, but also a lot of questionnaire behavior, looking at their cognition behavior and also at their anxiety, fear behavior, especially, and there's also an ADHD-like questionnaire. Interestingly, and that's what I didn't, because we see often in the clinic mainly really severely affected dogs, right? And they always have obviously the stranger-directed fear I told you earlier, and also to foreign dogs, and they also might be a little bit more anxious, right? But actually, they improved. And then, you should have written on the tin, because we obviously, as a specialist, we often see the worst of the worst. So when people used to use phenobarb in people mainly as an anxiolytic drug, right? So they were using an anxiolytic drug, and that was improving their behavior, right? And it's something we haven't really done, definitely in my career, right? And so it's actually what has been reported, but because the cases I always see are very difficult to treat, so it's probably not the drug which drives the behavior, it's actually the epilepsy itself which changes behavior, which makes sense to what I showed you earlier because of the brain pathology shown. Then the other thing is, surprise, surprise, we also looked at the microbiome and found some changes here in the Clostridia, but no big changes. But when it becomes interesting is when you look at the short chain fatty acid concentration, because what we found was that the dogs on phenobarb had an increase in total short-chain fatty acid concentration. And this is something, if you think about it, me as a neurologist would have never done this without these great collaborations here in Hanover with Gemma Mazzioli-Weber and Christine Effers, and then also obviously with Jan Sokodolski's group. And then when we looked at it, acetate also increased, but it was not, just 0.05, so it was not significant for the terms we defined before. And obviously, we found also that propionate and butyrate increased, which I thought was absolutely fascinating. But then what we did as the next step, we looked at, because we had some dogs becoming seizure-free and other dogs did not become seizure-free. And when you look at those guys, you can actually see that the responders had an improvement in butyrates and the non-responders didn't. Yeah, and this is interesting because butyrate is obviously used in intestine not only as a nutrition, so could it be another alternative energy source? It also has neuroprotective effect been demonstrated, and also is an anti-inflammatory component, has been also shown before. So really interesting, and something we have never thought before. So I'm quite excited about this. So the other thing is what we discussed earlier. We talked a little bit about stress and the interference, and we talked about other things, how this brain-gut axis can be modulated. And I remember when I came to Hanover, so four and a bit years ago, I came to Hanover, and I said, what we have to do, Jan? Why don't we see if we can actually also transfer behavior, because some owners report that the behavior they find worse than the seizures, right? And we looked in the literature, and that has been not so much out there for epilepsy, right? There have been, a person with Crohn's disease, as you know, FMT is one of the treatments you can do, which actually has a really good efficacy. But they have to go first through a battery of antibiotics, right? But also they have shown, obviously, that you can transfer behavior. It has been shown in rodents. People have done it from rodent to rodent. They have taken it from people with depression to rodents. So there has been a lot of different proof that behavior can be transplanted. So Anya here in her PhD used a protocol which we gratefully took, obviously, from the Munich group, and we are really grateful for you for this, guys, and obviously Jan's help as well. We looked again at behavior data and also looked at the brain shim data. Can we actually transplant behavior? And we had a donor dog which had no seizures. So it has idiopathic epilepsy, was treated with phenobarb, had no seizures anymore, and had really good behavior. Didn't have ADHD or fear and anxiety type of behavior. And already, what might be happening. Again, some mild shifters in the microbiome, and we found again some changes. But actually, the more interesting thing, I think, totally fascinating, was if you see on the right side the graph, you see that the donor's dog behavior in the ADHD impulsivity control. And you can see that actually what we have been doing by giving these 3 FMTs 2 weeks apart, we were changing the behavior of these dogs to the donor dog. So it improved the ADHD impulsivity control. Fascinating, right? And interestingly, when you look at the owners, yes, everyone will ask now about the seizure control. Yeah, it's like a small scale study. We had 10 dogs because we obviously, had to do a pilot study. So the authorities were happy with us. But, yes, some dogs improved in seizure control, but that was actually not the most important. The owners reported, and one has to be careful with the placebo because there was then no control, but all owners were happy that the behavior of these dogs improved, which we were proven not only by a questionnaire, but also in our brain shim as well. And that was really interesting. And also, obviously, certain non-social fear and chasing behaviors. We then looked at the neurotransmitter changes. And guess what happened? We had a mild decrease in glutamate, but a a really good increase in GABA. And the interesting thing there, this is measured in the urine, not in the brain. It's measured in the urine. And then the question is, how does a change in the periphery work to actually modify the brain, right? Because the blood-brain barrier is normally tight for these neurotransmitters. So it can't be a direct over the blood. So perhaps there's a different way of how it interacts. Drum rolls, I'm nearly finished. What we did then, we took some guinea pigs in science and looked at their with Jama's group, because they do a lot of Crohn's disease models and so on, and looked at the firing rate of the individual neurons. And the interesting thing was we took poop from dogs which were epileptic. We took dogs which were obviously not treated. Some were treated, responded, and then which were group C and group D were non-responders. And what we found was that the changes in the firing pattern. So you see on the right side, each neuron is depicted on the left side in these. So we had two things. On the one hand side, the burst frequency, and then also the firing pattern and which one was activated in this cluster of neurons, right? And the interesting thing was, at the end of the day, I would call it electric poop, right? So the dogs with epilepsy non-treated, they increased the burst frequency, but when we were giving drugs, it decreased again. So then we thought like, it's probably because phenobarb has a direct effect and changes this. But actually that was not the case. When we added phenobarb, I didn't have this now in here, but when we added, you have to trust me now, added phenobarb, it had no effect. Yeah, so it could be that it's secondary to the butyrate levels or not. So there's a lot more research to be done, and we have to do concentration curves. But the first indication is very interesting, because I was totally amazed that you had, you give this poop on it, right, from a dog with epilepsy, and then you change the firing rate. And that also could obviously explain the changes in your neurotransmitter profile. And perhaps what is happening is that the information gets transferred with the vagal nerve, because you have a change in the firing rate in those in the gut. Fascinating, right? People have done studies where they have given antibiotics, and some dogs improve. Some of this has been presented as an abstract. Same happens in people, but there the information is a bit sketchy. However, I will tell you just when I finish the story of Trudy. Trudy is a dog which was in our trial. Lovely dog, lovely owner as well. And the dog became very well controlled in seizures, perfect behavior. Owner was really happy. And then the dog saw my wife. And she's a dermatologist, as some of you might know. And guess what she did? She gave Trudy antibiotics, right? And guess what happens then? Trudy, no, I didn't get divorced, if that might be your opinion. I didn't get divorced. We still love each other, and I always will. But Trudy got worse. And actually, when they changed again, Trudy got better. So I think at least in one case, we could show the Koch's postulate. So I hope I could a little bit show you a top-down effect. I could show you also a bottom-up effect. I think there's tons of research to be done. How these all interplay. It's a totally fascinating thing. I never would have thought I do gastrointestinal type research. But because of all the work we have been doing, yes, anti-seizure medication is the mainstay when we treat dogs. Lifestyle might be also become important. Neurosurgery, neurostimulation will only be for a few, but MCT diet and nutrition has become a cornerstone also for seizure treatment at the moment, and not only for cognitive dysfunction. I hope you enjoyed the lecture. I hope you liked my little deviation into the gastrointestinal work, and thank you for inviting me. It was a big honor, and yeah, thank you.